the biological or the cellular foundation. In this aspect, we'll look at cytology and notable structural characteristics of cell. Look at a cell from the simplest to the most advanced. Now you look at it structurally, and you look at what are notable structural characteristics that are synonymous between cells. So we are going to compare and contrast features between the simplest of cell and the most complex, and we are going to explain observations. These observations, we are going to explain it to their structural and their functional level. We try as much as possible. So the cellular foundation of biochemistry explains that, first and foremost, cells of all kinds share some features such as plasma membrane. Now listen, let me take time to explain this. First and foremost is that cell of all kind share some structural features. For instance, plasma membrane. You notice that plasma membrane is seen in virtually all cells. You see your plasma membrane in virtually all cells. And what's the function of plasma membrane? The main function of your plasma membrane is that it chaperones, or it regulates what comes in and what goes out of the cell. It regulates what comes in and what goes out. It's just like a gate. In here you are going to have your plasma membrane. Your plasma membrane is formed formed as a result of conjugation of lipid and protein. Sometimes you also find carbohydrates on your plasma membrane. So it just regulates what comes in and what goes out. So what are the things that you see here? Now, the activity of plasma membrane on all cells is governed by, one, the availability of transport protein. For instance, here I may have transport protein. It is this transport protein that will regulate what comes in and what goes out of the cell. Another thing that you see on plasma membrane of all cells that regulates what goes in and what comes out is you see receptor. Yeah, you see receptor proteins. For instance, just receptors, they are just sensitive to some chemicals. So by the time they sense those chemicals, they are ligand gated. Yeah, in biochemistry we say those types of receptor are ligand gated. By the time the ligands now refers to the chemical that binds to them. So by the time those chemicals bind to them, those receptor proteins creates a crevice which opens up and allow molecule to either leave or enter the cell. And another thing is that we have membrane enzyme. Membrane enzyme could be on this plasma membrane to regulate what comes in and what goes out of the cell. So don't forget that I said it's a barrier that regulates what goes in and what comes out of the cell. And let me tell you a little about the characteristics of this plasma membrane. This plasma membrane is pliable. This plasma membrane also, like I said earlier, it's pliable, it's a metabolic barrier, it is hydrophobic, and selectively permeable. It does not just allow anything to go in and go out of the cell, it is selectively permeable. So having looked at this, let's look at one more structure that is general to living organism. Let's look at the protoplasm in the cell. The protoplasm. Now, the protoplasm is made up of cytoplasm and the nucleus. There is no, all right, so protoplasm is general to all cells. So here you see the cytoplasm and the nucleus. Now, the cytoplasm is composed of the cytosol. It's composed of cytoskeleton, It's composed of your inclusion. Now, so we also have vesicles. Now, here in your nucleus just contains the genetic material. Your nucleus contains the genetic material. It is also membrane bound, containing it's membrane bound, and it contains your DNA, your chromosome. It is the seat of your gene. Now, you notice that organisms, for instance, prokaryotes, your prokaryotes don't have a defined nucleus. The fact that your prokaryotes don't have defined nucleus does not mean that your prokaryotes don't have genetic material. They only have genetic material. Their DNA, or in some cases RNA, is lying. You see it. Sometimes it occurs in a circular form, sometimes in a single strand form. You see it lying in the cytoplasm. But in eukaryotes, we have a defined nucleus. So these features are general to all cells, and they are functionally active in all cells. Let's move on. The cytoplasm, like I said earlier, is that the cytoplasm is organized. The cytoplasm structure and the content of cytoplasm is not just there flying anywhere, that anything just move anywhere. No. I'm trying to say here, inside this cytosol, I have cytoskeleton. Now, this cytoskeleton, they help in maintaining the orientation and the organization of the cell. They are called cytoskeleton. You see them, they are composed of actin filaments, microtubule, and you have intermediate filaments. Yeah, you see them in virtually all cells. Now, these three, if you look at a cell under a very powerful microscope, you see them as strand. You see them as fibrous strand that is holding things in position inside the cell. Now, the fact that they are fibrous strand does not mean that they are static between the cell. Like I said earlier, they occasionally deform, they occasionally dissolve, and they reform, depending on the nature of the cell or the activity that is going on the cell. So another is that in the cellular explanation of biochemistry, 3 domains of life are recognized. In biochemistry, in the cellular explanation of biochemistry, 3 domains of life are recognized. We have let me explain it this way, life, which is divided this way. We have the prokaryotes, and we have the eukaryotes. Under the prokaryotes, they are divided into the bacteria and the archaea. So these 3 are the 3 domains of life. You have your bacteria, you have the archaea, and you have the eukaryotes. Now let's look at what these 3 are. Like I said earlier on, bacteria, they are the simplest form of life. They are prokaryotic. They don't have a definite nucleus. That's the main characteristics that's the main thing that characterizes them. They lack a definite nucleus. Now, archaea. Okay, let me explain this. Bacteria, let me take time to explain this. These bacteria, they are ubiquitous. You see them virtually everywhere. You see them on land, you see them in water, you see them in the air. Some of them are pathogenic. Now, moving to archaea. Your archaea, they are just like an intermediary between bacteria and eukaryotes. But the thing is that these archaeas, they live in extreme conditions. You see them in bogs, you see them in living under glaciers, extreme conditions that living organisms other organisms virtually adapt to. That's why you see actually now eukaryotes, they are organisms with defined nucleus. So that's just what characterizes eukaryotes. We have for instance, you and I, we are eukaryotes. So that's about that. Another thing is that organisms can either be anaerobic or aerobic. Organisms can either be anaerobic or aerobic. Now, they can also either be phototrophs or chemotrophs. What do I mean when I say an organism is anaerobic? Anaerobic organisms are organisms that can survive in the absence of air, in the absence of oxygen. Now, here when it comes to either being aerobic or anaerobic, aerobics are organisms that live in surplus, or whenever there is supply of air, they adapt well. Now, with these two being aerobic or anaerobic, there are now two categories. We have, they can either be facultative or obligative. Now, a facultative anaerobe are organisms that live in anaerobic condition and could also survive in aerobic condition. But when I say obligate anaerobes, these are organisms that survive only in anaerobic condition. They survive only in anaerobic condition. Now, moving to phototrophs or chemotrophs, this is telling us how organisms get their fuel and how they get their energy. How organisms get their fuel or energy. When organism gets its energy from the sunlight, it is said to be phototrophs. Organisms that get energy now, we will now have chemotroph. These are organisms that get energy from oxidation or reduction of molecules. Now, from here we now have autotrophs and heterotrophs. So we have to explain all this because they are fundamental to the knowledge of biochemistry, looking at it from the vantage point of biology. We have heterotrophs. Heterotrophs and auto. Now, autotrophs are organisms that can create their own fuel from simple elements of earth. For instance, your plants, they just get the sunlight and they integrate carbon dioxide with water. They do the redox oxidation and give you your glucose. Whereas heterotrophs, they cannot form their own food. They rather rely on already made food, already made fuels. Then they metabolize it, they catabolize it to create the energy they need. But autotroph, they create the energy they need for themselves using simple elements of earth and using energy from sunlight. That tells us something, that our phototroph then, let me show this sketch. Let's do it this way. Phototroph. My phototroph can either be an autotroph or an heterotroph. Now, my heterotroph, a chemotroph. Whereas my chemotroph, by the time I now have chemotroph. My chemotroph can always be an heterotroph. Now, my heterotroph can either be lithotrophs or organotrophs. Now let me explain this. My phototroph, which means organisms that has the ability to use sunlight as its form of energy, can be divided into either autotroph or heterotroph, which means it gets the energy from the sunlight. Now, having gotten the energy from the sunlight, it can create its fuel either by itself or it depends on already made food. Now, if it's depending on an already made food, then my phototroph is always a chemotroph, which means it is depending on already made food, which are chemical compounds on its own. So it's eating chemical compound. For instance, if I have a phototroph that will absorb glucose, it's a phototroph, but it's going to absorb glucose. Definitely, it's a phototroph, then it's an heterotroph because it is using glucose, which is a chemical, as its fuel. Now, looking at chemotroph, because my organism can be chemotroph straight, not having the ability to use sunlight at all to create its energy. Now, my chemotroph will always be an heterotroph. Now, after it's an heterotroph, it can now be subdivided into two. It can either be a lithotroph or an organotroph. When an organism is a lithotroph, it's getting its energy from metabolism of inorganic compounds. But if it's an organotroph, it's getting its energy from metabolism of organic fuel. All right, so let's move on. Another point under the cellular foundation of biochemistry is that all cells build supramolecular structures. All cells build supramolecular structure. What do I mean? What I'm trying to say here is that all cells has the ability to polymerize. What am I trying to say? Cells has the ability to bring together simple monomers of life. Then it condenses it together until it forms a polymer. This is what I'm trying to say. For instance, if I have glucose, my cell must have the ability to polymerize my glucose into glycogen. Now, if I have in my body, I have amino acids. Now, my cell must have the ability to polymerize my amino acids to protein. Now, from this protein, it even has the ability to condense protein into supramolecular structure, such as enzymes. So that brings us to the point of what we are trying to say. Our cells has the ability to bring together necessary simple monomers, simple biomolecular monomers. Then it brings it together. Then it condenses it together. After condensing it together, then at times it folds it up until it forms a three-dimensional functional structure. For instance, here I have amino acid. The amino acid can be tryptophan, alanine, valine, histidine, glycine, proline, whatsoever be it. Now, these amino acids, I won't go into the detail of the structure now because it's an introductory class. These amino acids are given amide linkage, one to the other, amide linkage. Then it forms oligopeptide. Now, my oligopeptide extends long enough to form protein. My protein is just a polymer which may contain as much as 10,000 amino acids. Now, this protein, after being formed, after being polymerized, my amino acids after being polymerized to protein can then fold. Here I have polymerization. The process here is polymerization. Now the process here is folding. So my protein here, after being formed as a result of polymerization of amino acid, can then fold to form a bigger structure, a bigger three-dimensional functional structure, such as enzyme. Sometimes it folds into hormones, such as you have in your catechemins. All right, now that brings us to this. We now have something that looks like this. I have monomers. Let me use this. I have molecules which forms macromolecules, and it eventually forms supramolecule. Now, molecule, for instance, here refers to my amino acid. Now, it gets polymerized to my macromolecule, which in this case is protein. Now, my protein can then fold into three-dimensional functional protein complex, such as enzyme, hormone, like I said earlier on. So this can fold. My protein can now fold together to form supramolecules, such as enzyme, such as hormone, such as organelles, such as membrane layers, and a lot of other things. So that's that about that. We just explained. So I'll go through the revision of what we have explained so far under the cellular foundation of biochemistry, that all cells share all cells share some kind of structural features. Under that, we mentioned structural features that cells share. For instance, we look at the plasma membrane, we look at the protoplasm and the content of the protoplasm. Then we look at the point that all cell is composed of cytoskeleton, which help in organization of the cell. We look at actin, filament, microtubule, and intermediate filament. And don't forget that we said I said earlier on that those filaments, they are just like fibrous strand. If you look at it if you look at a cell under a powerful microscope, and the fact that they look like fibrous strand does not mean that they are static. No, it is dynamic, because they are dissolving, they are reforming as the cell needs. For instance, during mitosis, these cytoskeletons, these cytoskeletal structures, they dissolve and they reform. During organelle migration, they dissolve and they reform. At times, this cytoskeleton, they also create a seclusion space for some cytosolic reaction to occur without interfering with the other activity of the cell. So the cytoskeleton are important. You see them in virtually all cells. Another point is that there are three domains of life. We have the bacteria, we have the archaea, we have the eukaryotes, like we explained earlier on. Another thing is that organisms can be divided based on how they use oxygen. They can either be aerobic or anaerobic, which on a larger extent, they can either be facultative, which means they adapt to both stances, or they can be obligate, which means they adapt to only one of the stances. And we also explain what phototrophs are, organisms that are phototroph. Why do we say organisms are phototroph? We explain their dependence on light as source of energy. We explain what chemotrophs are, that they depend on already made fuel as their source of energy. Then we explain the last but not the least, that cells has the ability to build supramolecular structure. How? They build it from simple molecules. They polymerize simple molecules into macromolecules, and they fold macromolecules into supramolecular structure. From there, we move to the chemical foundations of biochemistry. The chemical